in biology and mostly in medicine, we have those. I'm sure everybody heard MRI. How many of you have experienced MRI that is being pushed into the machine, having scans? How many of you? Raise your hands. Quite a few, including myself. So CT, that's another modality commonly used. How many of you have gone through CT scanning? Oh, quite a few, okay. PET, how many of you have used PET for yourself? Very few. Wow, that's amazing, nobody. Okay, so ultrasound, of course, is another one which mostly used to make sure the baby before born is okay, healthy. So maybe all of you have experienced that. Of course, optical imaging, that is very commonly used in biology, especially with living cells. So talking about MRI, MRI actually is essentially a part of nuclear medicine. It's to use, as the name suggests, use magnetic resonance as a way to visualize different organs of the body. So as you may know, actually 2 pioneers of the MR studies got Nobel Prize in 2003, actually one from UIUC. So essentially, the way MR works is to actually apply a pretty large magnetic field. Because in the body, we have a lot of water, right? If you take a cell out, it's essentially mostly water. So when you apply a very large magnetic field that aligns the water molecules, especially the protons, as a spin, as a dipole, and that spins when you have a radio frequency wave sending to the body to disturb the equilibrium, then you stop that. So there's a relaxation process that restores the equilibrium of the whole field. So that's actually how it's so-called magnetic dipole moment. Right? Essentially, it is that the decay of that vector field gives you the signal in MRI. So that's basically how it works. So as a result, so far MRI is mostly effectively used in imaging soft tissue because that's where you have a lot of water molecules, right? So that's the first imaging modality, of course, widely used in medicine. So the second one is essentially X-ray. It's the computed tomography, or called CT. So that one, the difference between CT and MRI is that, of course, this is based on X-ray, right? Also, it is more effective in imaging hard tissue instead of soft tissue. That's why usually what you see in a CT scan is mostly bones, right? As you can see, this cross-section of a bone. You see clearly the structure of the bone. So that's actually how the X-ray images more effectively tells us it's really the hard tissue that we try to image with CT. But if you combine the two, of course, you can have a clear idea of the morphology of the hard tissue and the soft tissue, right? So the third one is just going through quickly is positron emission tomography, or PET. So that one, it is not by tissue itself to generate an image. You need to have something else as a radio tracer that actually emits the photons. Basically, for the mechanism, you have a radio tracer such as F18 or copper-64. And if you inject those radio tracers in the body, then that radio tracer could emit photons actually roughly with an angle of 180 degrees. Okay, so then you have a device that detects those photon emissions and then recalculate based on the information to calculate where the radio tracer is. Okay, then using that to construct a image. So, the three-dimensional images of the radio tracer could reflect specific functions of the biological process in the body. Okay, so in a way, although MRI and the CT are mostly on the morphology of the tissue, PET is mostly on the functional states of the tissue. So that's a major difference between PET and the other two. Okay, so of course, as you know, PET is more expensive. That's why very few people in the room have done PET, because usually, unless it's needed or the insurance company agrees to cover, otherwise you will not have PET. Okay. So, but it's very, very useful because compared with CT and MRI, PET is more sensitive. It's roughly 100-fold more sensitive than MRI. Okay, so that's why in a lot of studies we use PET. So ultrasound, as I mentioned quickly, of course, is well known. A lot of different applications. In addition to evaluating this, the health of the baby or unborn baby, another very important application is in cardiology, so-called ultrasound cardiography or echocardiography. So of course, I would assume quite a few of you have used this, or the doctors have used this to evaluate the health of your heart, right? So that's actually mostly used in hospitals, in clinics. If you compare those, here we have two scales. One is the spatial dimension, okay? So from 10 to minus 13 all the way to 10 to 1. So that's meter, right? So you can see that the PET, so this is the range of PET, CT, optical, which is the visible light, and MRI, which is here. So, and this is actually more like a time domain, which is in terms of frequency, okay? So you can see that different imaging modalities covers different ranges in terms of both the spatial dimension and the time, okay? So the good thing is that in many cases you can combine those. For example, you can easily combine PET and CT, like Siemens and other companies have produced imaging machines or scanners, which is modular, so that you can easily combine two modules. One is for PET, the other for CT, and you can use CT to visualize, as I mentioned, the heart tissue, especially bone, to give you morphological information. Then you can use PET to give you functional information, right? Combining the two, you could get a much better idea of the state of the tissue. So there is also an effort to combine PET with MRI. Okay, so as I mentioned, MRI is mostly for soft tissue. And in that case, it's the same thing. You can, if you have a machine combining MRI and PET, what you can do is you can use MRI to give you the morphological information, mostly for soft tissue. Then you can use PET to give you the functional information. So this together, of course, will tell us a lot about the state, again, the state of the tissue, the soft tissue. Optical imaging, it is widely used in visualizing what's going on in cells. It is light-based, so if you try to get images in deep tissue, you actually have some limitations because whatever light you use, the depth of penetration is very limited, right? So if you try to visualize a specific part of the organ, you need to have light penetrating into the tissue, and you need to get signals out, right? That's why for deep tissue imaging, optical method is quite limited. So that's why typically we use PET, CT, MRI, or ultrasound for deep tissue imaging, and we use optical imaging mostly for cellular studies. So this is another comparison of the different imaging modalities. So CT, ultrasound, and MRI, those three are mostly for morphological or physiological information, okay? So PET could give you some functional information. So optical imaging, of course, could give you information at the cellular and the molecular scales, right? So PET, since we use radio tracer, which could be a function, could be a reflection of the local events, molecular events. So in a sense, you can also use PET to get some molecular information. So that's really in terms of the applications. Today I will mostly focus on optical imaging methods. I heard from Roger that you have constructed microscopes. How successful you were? Very successful? I heard you will use your microscopes to do further studies later in the week. Okay, good luck. Okay, we'll see. So, as I mentioned, if you want to visualize cells, of course, the optical imaging is most useful because of all the imaging modalities, optical imaging gives you the best spatial resolution. So since you have built a microscopes, what is the spatial resolution you can reach with a typical microscope? Any idea? I cannot hear you. What's the spatial resolution you can reach with an optical microscope?
200 nanometers.
It's a 250 nanometers, okay? So actually it is here you could easily answer the question. So anyway, so that's why if you do cellular imaging, because of the spatial resolution, it could give you very detailed information of a cell, okay? It is still not to a single molecule scale because typically a protein has a size of roughly 10 nanometers, right? So it's still, so the spatial resolution of 250 nanometer is still much larger than the size of a single protein. But what we really want is to visualize protein-protein interactions, okay, in a cell. So to do that, you really need to have a much higher spatial resolution. So that's why in recent years, a lot of efforts have been made to develop so-called super resolution microscopes. Okay, there are a few of those. One is called STAT4Pi. The other is called STORM. We also have so-called PALM and others, so-called structural illumination. That's another one. So those microscopes could reach much better spatial resolution, like STAT4Pi, the inventor, Stephen Hell. He claims that you can reach roughly 30 nanometer spatial resolution. Okay, it's much better than 250. So some of those are being commercialized. I know the STAT has been commercialized by Leica. I heard STORM is also being commercialized. definitely palm and the structural illumination have been commercialized. So nowadays, you can actually buy a microscope with a much better spatial resolution using one of those techniques. So let me just give you some idea of the storm, which was developed by the MIT's neighbor, someone at Harvard, Xiaowei Zhuang. She actually was a postdoc at Stanford with Stephen Chu. So the storm actually uses the photo-switchable fluorescent probes, such as Psi3, Psi5. So essentially is to temporally separate the otherwise spatially overlapping images of individual molecules. Then from that, you construct the images to reach higher resolution. So just to give you an example, if you try to image microtubules, the conventional microscope gives you a very blurred image. Okay, it's because, of course, you do not have the necessary spatial resolution to image individual microtubules. Now, if you use storm, since that can reach roughly 20 nanometer spatial resolution in the XY plane, you could actually see much better images of microtubules, right? And if you compare this with that, of course, it's much better, right? So as I mentioned, it has been, at least I haven't seen the machines yet, but I think it's being commercialized. So using that, you will have to, in addition to the machine itself, you will have to use the right dyes to form a pair. Actually, I believe initially Xiaowei Zhuang used the Psi3, Psi5, okay? You have to use the right pair of fluorescent dyes in order to get the better spatial resolution in storm. In the rest of the lecture, I will first quickly mention some challenges in developing probes for imaging proteins in living cells. I will focus on probes for live cell imaging of RNA molecules. I will also give you some ideas of the in vivo imaging probes. So for proteins in the living cell, as I mentioned, ideally we would like to see not only where are the proteins, but also how proteins interact with each other. So Peter Wang last Friday, I'm sure, gave you a great lecture on using FRET, using the resonance energy transfer between two fluorescent proteins as a way to visualize protein-protein interactions. That's definitely a very nice way to do so. So, but in a sense, if you want to see either just one type of protein or two proteins, you have to tag them, right? So the question is how to see proteins in the living cell. So different tags have been developed, such as fluorescent proteins, flash or RIAs, SNAPE, HALO, and so forth. So all of those actually use something to tag the molecule. In many cases, in many cases, fusion proteins. So once you have a tag, of course, since it is directly on the protein you want to study, it has very high specificity because that's exactly what you have. The problem, of course, is that usually in doing so, you have to transfect the cells. In that case, of course, you would alter gene expression, and that's perhaps biology. And in some cases, the tags are pretty large that actually could also influence biology, okay? So if you want to directly label proteins by delivering some fluorophores, That, of course, does not require like transfection, right? So you will not really alter the expression of the specific protein, but you also have issues because you need to deliver them into cells. Deliver itself could be an issue, right? And so that's why, for now, most of the studies directly using fluorophores or quantum dots is to really target cell surface proteins, because that way you could avoid delivering the fluorophores into cells. But for intracellular imaging purposes, you have another issue. If you deliver a dye into a cell, a living cell, and after delivery, you cannot wash the dyes out, right? Because the cells would not be happy if you try to wash things out of the cell, right? So as a result, whatever you deliver into the cells, they give you signal, right? So that's a major limitation, because in that case, doesn't matter if the specific dye binds to the protein what you want to study or not, they give you signal, right? So that's background signal. So that's actually a major challenge. So there's still a need to develop a method that could specifically label proteins in a living cell without changing the expression, right? So as I mentioned, the first method, you typically alter the gene expression or protein level. The second method, you have the issue of high background signal. So ideally, what you should have is a probe that binds to unmodified proteins. Only when the probe binds to the protein, it gives you signal. Otherwise, no signal, right? So that way you avoid changing the expression level. You avoid a high background signal. So essentially, that's one of the projects in my lab to develop such a probe. I'm sure you have heard fluorescent proteins, which is the mostly used fluorophores in biology studies, because many, many labs, as you heard from Peter Wang, use fluorescent proteins as a way to tag specific proteins as a fusion. So it's mostly due to Roger Tian's efforts. Of course, he's a professor at UC San Diego. He received 2008 Nobel Prize for his work in fluorescent proteins. It is actually, again, due to Roger's effort, it is multicolor. You can see from blue all the way to red, to dark red. So you could realize all kinds of colors in between. And of course, that gives us a lot of abilities to visualize different proteins, also to do FRET, as you heard last Friday. Could you close the door, please? Thank you. So I briefly mentioned quantum dots. I would assume at least some of you have heard quantum dots. Those are nanocrystals, typically semiconductor nanocrystals. It's very bright. Typically, it could be at least 20 times, up to 100 times brighter than typical fluorophores. It's very photostable. Essentially, there's no photobleaching. And the good thing is also that you can use a single excitation wavelength, typically between 300 to 400 nanometers, to realize different colors. You can see it's also multicolor. Of course, over the last 10 years or so, a lot of labs are using quantum dots for biological studies. So one type of studies we have been performing is to try to understand the assembly and disassembly of two major molecular machines that repair DNA double-strand breaks. One is called NHEJ, which is non-homologous interjoining. The other is a homologous recombination. So each pathway or a molecular machine consists of different proteins they assemble at the brick site. So the cells have this amazing ability to repair DNA double-strand breaks very quickly and very precisely. So the amazing feature is that once there is a DNA double-strand break in a cell, either due to the food we eat or due to the light, especially UV, so once there is a break, the protein complex quickly assembles at the break site, then fix the break. Okay, so it typically fixes the either way, either with NHEJ or HR, quickly repairs all the DNA double-strand breaks. So we try to understand how that happens. So there's also an interesting biological issue that when there is a DNA double-strand break, how cells decide which pathway to use. It's so-called pathway choice. So a lot of interesting issues, but the critical thing for us is to visualize the assembly and disassembly of those machines. So I just show you one example is to use fluorescent proteins, different fluorescent proteins such as ECFP, EYFP, M-cherry, and M-plum, to use different fluorescent proteins with different excitation and emission wavelengths to tag different components. In this case, NHEJ, like we use ECFP to tag 53BP1, use YFP to tag SR64, and to use M-cherry to tag Q80, is to use fluorescent proteins to tag different components, then trying to visualize when those components assemble into the protein machine, how quickly they assemble and how quickly they disassemble. So just to show you one example. There are different, as I mentioned, different ways to tag intracellular proteins, but there are pros and cons. So as I mentioned, for example, if you use fluorophores, the good thing is that they are small and you have many choices. You can choose the dye you want. The big issue, I think the biggest issue is actually the photo bleaching, because if you try to follow some dynamic processes in a living cell, and you need to visualize the process for, let's say, a few minutes, for example. But typically, the fluorophores quickly photo bleaches. Actually, some of the dyes you use, or we typically use, the fluorescence intensity can be reduced by 90% within 1 minute, right? Imagine you want to follow the process by 10 minutes, and the photo bleaching will kill the study because once you pass just a few minutes, no signal, right? So that's actually a big issue. Of course, the other issues such as the hydrophobicity will prevent you from delivering them into cells. So if you use fluorescent proteins, as I mentioned, the good thing is that it's highly specific, but the problem is, again, photo bleaching, and also, in many cases, the signal level is pretty low. If you use quantum dots, you could have very high signal level. Also, you could have multicolor imaging because, as I mentioned, you can use different quantum dots with different emission wavelengths, and you can excite them with a single wavelength. But the size is the problem because usually it is larger than 20 nanometers. And the protein you want to tag, let's say it's 10 nanometers, the probe itself is larger than the protein you want to study, right? It's a problem. Especially if you want to use quantum dots to label different components of a molecular machine, and the size of the quantum dot could interfere with the assembly. Okay, so there are different challenges. So then this is more overview of the fluorescent probes for live cell RNA imaging or detection in living cells. So this actually was part of a review I wrote. I believe you have received a review article we wrote a few years ago, roughly 2 years ago. So anyway, so for messenger RNAs or any RNA in a living cell you want to image, of course you have to use the probe. So the simplest probe for RNA detection and imaging is actually, see, let's see, this is the RNA molecule you want to, a specific sequence of a RNA molecule you want to image. The simplest way is just to take the complementary sequence, which is a linear oligo, then you attach a dye, right? So when the probe, of course, binds to the target, you have this situation. So the problem is that if you use this probe, you deliver them into cells, as I mentioned, doesn't matter it binds to the specific RNA molecule or not, it gives you signal, right? So that means the background signal is quite high. So then, of course, you can use two of them. Both are linear oligos, one with a donor fluorophore, the other with an acceptor fluorophore, right? So in that case, you have two probes binding to the same target RNA, and then you use the fluorescence resonance energy transfer between the two dyes. I'm sure Peter Wang explained the idea of FRET, right? So it's basically use the idea of FRET. So this, of course, by detecting FRET signal, you could have a much better specificity in terms of having the positive signal, but you could still have some high background signals due to non-specific binding, because the linear probes, let's say if you have a 20 base target domain, even if you have roughly 15 bases or 12 bases that are complementary to the target, it can still bind. So in some cases, you could still have a pretty high background signal due to non-specific binding, right? Then comes to the idea of having a molecular beacon, which is a hairpin probe. So you have a loop portion which binds to the target. Then you add two short sequences to form the stem. So the stem, at the end of the stem, you have a dye and a quencher. So the quencher quenches the fluorescence of the dye initially, right? So that's why I mentioned if you directly deliver a probe into a living cell, since you cannot do washing intracellularly, once you deliver the probe, it gives you signal. Doesn't matter it binds to the molecule you want to study or not. Now for molecular beacon, it has this nice feature that if it does not bind to your target, in this case RNA molecule, it has no signal because the fluorescence is quenched by the quencher, right? So, but once it binds to the specific target you want to study, in this case RNA molecules, it opens. Actually, the basic idea is biomechanics in a sense. That is, you have a few bases here, usually 4 or 5 bases, either 4 bases or 5 bases. So you have enough energy to form the loop, right? So that way you form the loop, you have the stem, you bring the dye and the quencher together to quench the fluorescence. But once the probe binds to your target, it becomes double-stranded. So let me ask you, if you have a DNA molecule, if you study the mechanical property of DNA molecule, what is the difference between the binding rigidity of a DNA molecule between single-stranded and double-stranded? What's the difference between the two? How many fold of difference if you compare the binding rigidity of single-stranded and double-stranded? Any idea? It's 50-fold. Okay, so that means in order to bend a single strand, it's very easy. I can easily form a hairpin, which is this, right? But once it becomes double-stranded, since there's a 50-fold increase of bending rigidity, you need to have a much higher energy in order to form the loop, right? But you do not have enough energy here. So that's why it opens, right? So once it opens, the quencher is separated from the dye. Now you have fluorescence. So this is the kind of probe we want. That is, before it binds to your target, no fluorescence, right? Only when it binds to your target, it shows fluorescence, right? For intracellular imaging of biomolecules, either a RNA molecule or a protein, this is exactly what we want. That is, we want to have a probe. It does not give you signal when not binding to the target. It gives you the signal if there is a binding event, right? So since you cannot do washing, this would give you a more specific signal. So that's the basic idea of a molecular beacon. Of course, sometimes when you deliver beacons into living cells, enzymes could actually chew them up. So once you have degradation of the probe, you could still have high background signal because the dyes then would give you the signal, right? So then we actually, in parallel to this, we can use two molecular beacons. One has a donor dye, the other has an acceptor dye. Then in this case, of course, only when both beacons bind to the target, you have signal. Otherwise, you have no signal. So since in this case, you have the beacon format of the probe, typically if there's no binding, you have essentially no signal. And even if the beacons are being degraded, you will not have FRET signal because individual dyes will not give you FRET. Right? So that's the beauty of this assay format. Then there are some other alternatives using ligation, in this case, using so-called MS2, which is really a protein, the so-called MS2 coat protein, binding to hairpins. Then you use a fluorescent protein to tag the MS2 coat protein, right? That's another. So this is actually very novel but very difficult. That is, you basically have two halves of the fluorescent protein. Then when you bring in the two halves of the fluorescent protein to close proximity, then you actually reconstitute the fluorescent protein. But actually, it's not that easy. So even if you are successful, this whole process may take up to 2 hours. Imagine if you want to study the dynamics, then this would be too slow for your purposes. Anyway, so just give you an overview of the fluorescent probes for RNA detection. So delivery actually is an issue. Yes? I'm sorry?
How long is the degradation of the...
Oh, the degradation. Okay, that's a great question. So for degradation, really depends on what is the cell type. Okay, so based on our experience, for example, if you deliver molecular beacons into living cells, for cancer cells, since in cancer cells the enzymatic level typically is high, right? You could have beacon degradation as short as after 40 minutes. In normal cells, usually degradation occurs after 2 hours. So that's really the window of your imaging assay, right? Yeah, that's a great question.
And how does it cascade the signal?
Right, actually, that's the next slide, yeah, which is here. Okay, so that's another good question. So how to deliver beacons to pass through the cell membrane? So we actually developed 2 methods, and in a way 3, but this is the combination of the 2. So basically, the first method uses a toxin called the streptolysin O, or SLO, SLO. So what the toxin does is to regroup the cholesterol molecules on the cell membrane to form a ring-like thing. And inside the ring, it's a hole. Okay. The hole actually typically has a size of roughly 30 nanometers. Molecular beacon itself has a size roughly 5 nanometers. So once there are holes on the cell membrane, the beacons would diffuse in. Okay, so that's one of the delivery methods. The second method is actually to have a peptide called cell-penetrating peptide, such as TAT1 peptide or polyarginine. So basically what we did was to conjugate a peptide to a single molecular beacon. Then the cell-penetrating peptide could carry the beacon into cells, going through the cell membrane. Okay, so a few years ago, we wanted to image nuclear RNA molecules, that is RNA molecules in the cell nucleus. What we did was to conjugate a nuclear localization signal peptide to a single molecular beacon, then use the slow-based method first to deliver this complex into cell cytoplasm. Then the NALS peptide would carry the beacon into cell nucleus. So you can use either slow or the peptide or the combination of the two. Okay. So that's the delivery. Let me quickly go through some of the design issues. If you want to design a molecular beacon, what you can do is first you know the specific sequence, such as surviving messenger RNA sequence. You get a sequence, then you select unique sequences, roughly 15 to 20 bases long, and you can use the bioinformatics tools to actually search and identify the unique sequences, or you just choose a sequence, then you do a search to make sure that it's unique to the gene you want to target, right? So once you have that, usually it's fairly easy to identify those. Once you have that, you add a stem sequence, roughly either 4 or 5 bases. You add the short sequences to the end of your target sequence to form the hairpin. Then you can actually order beacons. Usually what we do is to order molecular beacons from a company because it's cheaper that way. You can, of course, synthesize your own molecular beacons, but actually takes more time and more expensive. Okay, so that's why we usually do not do that. But typically, once we have the beacons from the company, we do our own studies to make sure that the beacon the synthesized has a pretty high signal-to-noise ratio. Usually, the signal-to-noise has to be larger than 10. Okay, otherwise, it won't be so useful. Then you do delivery, as I mentioned, just to give you some idea of the localization. So in terms of cellular imaging, you can see for surviving messenger RNAs in prostate cancer cell line MIA-PACA2, pancreatic cancer cell line MIA-PACA2, you see the signal is on one side of the cell nucleus. That actually shows where the messenger RNAs of survivin localizes. For beta-1 integrin messenger RNA in MG63, which is the bone cell line, I believe, and you see the signal is roughly at the paranuclear region, that is right outside the cell nucleus. And for androgen receptor messenger RNA in a prostate cancer cell line, LANCAP, you see that the signal is on the edges of the cell. Okay, so this actually is an interesting biological issue. That is, number one, if the localization of the messenger RNAs is due to functional requirements, right? For example, for some messenger RNAs, we found that they localize with mitochondria, okay? And there are some functional relations between the specific messenger RNAs and the mitochondria. So the other question is if the localization, especially in cancer cells, if that reflects the disease state of the cell, right? So those actually are important questions in terms of biology. So one specific issue in terms of engineering design is that you need to be careful in designing a molecular beacon, because even if you identify specific sequences of a molecular beacon, it may not give you signal. The reason is that you never, in a living cell, you never have just the messenger RNA. So the messenger RNA molecule is always complexed with RNA binding proteins. At different stages, you always have proteins on messenger RNA. So therefore, if you design a beacon, let's see, it so happens that the beacon sequence or target sequence for the beacon is roughly here, and you have a RNA binding protein sitting on the sequence, then that's bad luck because the beacon will not be able to bind because it has to compete off the RNA binding protein, which could have a pretty high affinity, right? So also in terms of the so-called secondary structure of the messenger RNA, you could have a portion that has double-stranded, okay? And if you have a beacon, you select a sequence which is roughly here, also you would have difficulties because it's already double-stranded, right? It's difficult for the beacon to bind. So ideally, you would select a sequence somewhere here. There's nothing there, then the beacon could bind. So that's a specific design issue. Just give you some idea of the recent applications we have using molecular beacons. Also, this is actually related to GEM4, to the EBICS studies. I'm pretty sure you heard about EBICS, right? Which is the center we have with a lot of efforts in developing cellular machines. So we have been using molecular beacons in stem cell studies. One type of study is to actually detect and isolate cancer stem cells or iPS cells. For example, if you try to convert skin cells into iPS cells, very often the efficiency is very low. That is, cells that are successfully being converted into iPS cells, the percentage is very low. Okay, it's I heard it's less than 0.01%. Okay, so that means you have a lot of cells, but very few of them are really iPS cells, and you can use beacons to detect them. So that's one possibility because usually the surface markers are very limited. So you want to also keep cells alive. So we can use beacons to target genes that are highly expressed in stem cells, such as OCT3, OCT4, SOX2, and Nano. Okay. The other thing which is also important for the EBIC studies is to isolate a specific subtype of a cell, because once you have stem cell differentiation, usually you have quite a few cell types all mixed, right? If you want to select a specific cell type, such as cardiomyocyte, and there's no good way to do that, but we can use molecular beacons to do that, because again, you can target a specific messenger RNA that is highly expressed only in that cell type, right? Then you do flow cytometry with beacons to do that, to select the cells. So just give you some idea, in this case, we designed a beacon to target OCT4 messenger RNA in cancer stem cells because OCT4 is highly expressed in all stem cells. And this is just to show you, before differentiation, let's see if the normalized level of OCT4 messenger RNA is 1, after differentiation is almost 0, okay? So it's reduced by more than 95%. And this is a comparison of the protein of OCT4 in stem cells and in differentiated cells, okay? So we use this feature, designing a beacon. You can see that that's the red signal is the signal from the molecular beacon we designed targeting OCT4. This is in stem cells. This is in differentiated cells. After differentiation, as I showed you, the OCT4 level is very low. That's why actually you have a very low fluorescent signal. Now, if you use a dye to visualize all messenger RNAs, there's no difference between stem cell and the differentiated cells, just to show the specificity. So what we did was to isolate the cells using flow cytometry. Actually, in this case, just to be sure, we also used a dye-labeled antibody to target SSEA1 protein on the stem cell surface, the red signal actually shows the signal from dye-labeled antibody targeting SSEA1. So if you have both probes, now in this case you have undifferentiated stem cell and the cells after differentiation, you have no probe at all. Then you perform flow cytometry assay. There's essentially no difference. Okay, you see the contours, essentially they are the same. You cannot tell any difference, right? So now if you use both probes, the x-axis is the beacon signal, the y-axis is the signal from dye-labeled antibody targeting SSEA1. Then you have very different contours. So those contours are from stem cells. Those contours are from differentiated cells. Okay, so using flow cytometry, you can actually isolate the stem cells because you see large differences between these two populations. Of course, if you mix the two populations, you see mixed contours. So one issue, especially biologists often ask us, if you deliver molecular beacons into stem cells or other cells, trying to isolate them, what are the consequences of doing so? Because the probe itself may influence, for example, stem cell biology. So we checked that. So what we did was to have two assays with stem cells, one with beacons, one without beacons. Then we measure the messenger RNA level of OCT4 because that's really the beacon target, right? So OCT4 is the target of the beacon. You see no difference because this is with beacon, this is without beacon before differentiation. That's in stem cells. After differentiation, again, with beacon, without beacon, there's not much difference, essentially the same, okay? So we took another gene, which is IGF-2, which has the other trend that is before differentiation, the expression level is very low. After differentiation, the level increased by 150-fold. Again, with beacon and without beacon in stem cells, essentially the same. After differentiation, with and without beacon, again, it's essentially the same. So, yes?
What kind of beacons made of DNA or RNA?
That's a good question. So you could use either DNA or RNA, right? So the RNA molecules, if you use RNA molecules to form the backbone of the beacon, the affinity actually is higher because you would have RNA binding, right? With DNA, usually the affinity is lower because you have DNA RNA binding. So depending on the purpose, because for some studies, the difference would be small, and usually DNA beacons, we call DNA beacons, are much cheaper, right? But if you want to make sure that the affinity is high enough, right, and you actually choose to use RNA beacon or to use RNA backbone, right? Yes, that's a good question.
With the length of the beacon, do you ever have any problems activating like siRNA related?
That's another great question. So as you know, the siRNA molecules usually has a length of 19 to 22 bases, right? And the beacon actually usually is right in that range. It's usually between 15 to 25 bases, right? So we actually have done studies to show if you use, actually that relates to your first question. If you use DNA beacon, then what you have is when the DNA beacon binds to your RNA target, you have the RNA-DNA complex, which will not induce siRNA effect, right? Now, if you use RNA binding, in theory, you could. Okay, you could. But our studies have shown that the chances actually are not that high, okay? Although we cannot see absolutely that there's no effect, right? So that's another reason for some studies you want to avoid potential siRNA effect, you use DNA beacon, right? But both are great questions. So in any case, in this case, we were convinced that the beacon itself will not really affect stem cell biology. How many minutes I have? 5.
Yeah, that's plenty.
Okay, yeah. So let me quickly go through another probe in addition to beacons. So one thing we have done recently was to combine fluorescent proteins, which is multicolor, with quantum dots, which is also multicolor, as I showed you. So what we did was to conjugate fluorescent proteins, multiple fluorescent proteins, to a single quantum dot through a linker. Okay, so that way there's a fluorescence resonance energy transfer between the two, right? So the linker actually serves multiple purposes. One is to bring the fluorescent proteins to the quantum dots so that you could have FRET. The other purpose is that you could have part of the linker as a substrate of enzyme, a specific enzyme. Then in that case, if you do have enzymes, the enzyme would cleave the linker, the fluorescent proteins would go away, and you will actually eliminate the FRET signal. Okay, so that's, in that case, you really have a enzymatic activity assay, which is essentially the first application, that is to have an assay to quantify enzymatic activity. The second application we explored is to actually use the pH sensitivity of specific fluorescent proteins, then combining that with quantum dots to have a probe for quantifying intracellular pH. So let me just focus on the second application. Let me skip that. So in that case, if you just take M orange, which is a fluorescent protein, and those are the absorbance of M orange, which of course is pH sensitive, you see the different curves with different pH values from roughly 5.4 to pH 10. Okay, so you see different absorbance. And if you plot that, you see this quite nice curve. Okay, this is actually the M orange molar extinction coefficient as a function of pH. So we combine that with quantum dot to form a FRET probe for pH sensing. We use quantum dot 520. That's the emission peak with M orange. Again, we have this very nice set of curves for absorbance with the same pH range, that is from pH 5.4 to pH 10. If we plot the acceptor emission, which is M orange, versus donor emission, which is the emission from quantum dots, we have this very nice curve. The good thing is that the pKa is roughly at 7, and also from pH 6 to pH 8, we have 20-fold increase of this ratio. That means actually it's quite sensitive in this case. Then we did studies. This is actually important. That is, I mentioned photobleaching is the issue with many dyes and with fluorescent proteins. One dye that is commonly used for pH measurements is this dye, BCECF. You see that the green curve, that is within 1 minute, the fluorescence intensity of this dye is reduced by more than 90%, okay, within 1 minute. So if you take the M orange protein, and which is the red curve, within 2 minutes, the fluorescence intensity is reduced by more than 80%. Okay, so photobleaching is a big issue. Now, if you combine the M orange with quantum dots, even after 10 minutes, you still have 50% of the signal. So that's one of the major reasons why we want to combine M orange with a quantum dot so that we could address the issue of photobleaching. The other major advantage is the fold difference in the range of pH 6 to pH 8. this is actually a comparison, the fold difference. With our probe, the fold difference is, as I mentioned, roughly 20-fold. With just M orange, actually it's much lower. It's roughly just within the range of 5. Okay, so that means this probe could be much more sensitive. And we did some intracellular studies to deliver the probe into HeLa cells. The idea is that the probe goes through this endocytic pathway. In the endocytic vesicle, pH is roughly 6.5. In the early endosomes is roughly 5.5. Late endosomes is 4.5. Okay, going through this, you should see different levels of Fred signal. And with low pH, the Fred signal should be very low. Indeed, it's the case because we deliver the probe into cells and image the cells right after delivery. You can see that right after delivery, you see pretty strong Fred signal as in blue. So if we wait for 2 hours, and in that case, all the probes should be in late endosomes, and indeed the Fred signal is essentially gone. Okay, so that's indication that the pH probe worked in living cells, and this is just one of the controls. We use drug molecules to arrest the endocytic process, right? So therefore, the vesicles will not move into late endosome because of the drug molecules. In that case, this is right after delivery. You have strong Fred signal. After 2 hours, you still have Fred, very strong, roughly the same level of Fred signal, right? So that's an indication that the probe worked. I guess I just have a few minutes left. Let me quickly mention the other type of probe is really to use nanoparticles for in vivo imaging, to have a probe for MRI, for PET, or for combined MRI and PET. I just want to show you the idea that if you take iron oxide nanoparticles, which is a MRI contrast agent, it's a T2 agent. So you coat the iron oxide nanoparticles with biopolymer. In our case, it's phosphorylated PEG. And on the coating itself, you could have quite a few things. You could have a dye, because in some cases, you want to, after you perform in vivo imaging, you want to look at the tissue sections. Then you can use the fluorescent dye to do that. Or you can put a radio tracer such as F18 or copper-64. In that case, this becomes a PET contrast agent, right? So as I mentioned, now we can do combined MR and PET, and this is a contrast agent for combined MR and PET because you have the MR contrast agent, you have the PET agent, right? So you can do both. And of course, you can have a targeting ligand on the surface of the nanoparticle. This is just to quickly show the as synthesized nanoparticles. We can control the size very well. You can see this average size roughly 6.4 nanometer, 10.4 nanometer, 14.9 nanometer, very uniform. Okay. So then we can coat them with the coating, that those are the iron oxide nanoparticles, and we can use the same coating for quantum dots. Let me skip some of the details just to show you. We can functionalize them because in doing the coating, you can use functionalized PEG. So then we can actually conjugate half of the antibody to the surface of the iron oxide nanoparticle as a way to do targeting. Of course, we can also conjugate targeting peptide for specific cell surface receptors. Let me skip all that. So this is just to show, let's say if you want to target tumor cells, one of the target is Folate receptor. So we can conjugate an antibody to target the Folate receptor tumor cells. And this is just to show with fluorescence dye on the iron oxide nanoparticles, you can quantify the targeting events because you see with two different cell lines, this has high expression level of folate receptor, which is actually healer cells. You see strong signal. And this is another cell line, 435S, which has very low folate receptor. You have very low signal. Then we can quantify that. Then to do imaging, we actually generated a tumor here. Then we performed MR imaging. Typically, you take a series of images, then construct so-called T2 map. In fact, the 1 over T2. Without any nanoparticle, you see in the tumor region, that's the T2 map. Okay, so the higher the contrast, the more red you should see. And indeed, with nanoparticles, you see a lot more red in the tumor region compared with the situation without nanoparticles. So in terms of MRI, the contrast can be increased dramatically. And we still have room for improvement because in this case, you can see the red signal is mostly on the near the edge of the tumor. And ideally, we would like to see the whole tumor section becomes red, right? So we still have challenges, but I think it's doing. The probe definitely works. So with that, I would like to acknowledge my lab members and collaborators on the results I showed you. Alison was a PhD student. She's doing a postdoc now. And David was an undergraduate student doing the quantum dot fluorescent protein probe development and also some of the pH sensing work. He is now a PhD student. Wang Jiang was a postdoc in my lab. He did the live cell molecular beacon studies and the imaging of pH in living cells. Shen and Annie, Shen is a postdoc, Annie is my lab manager. They did most of the work of the MRIs I shown you very quickly. Bill Danny is a collaborator. I shown you how to use fluorescent proteins to tag different components of the DNA repair machine. That was from Bill. And those are the grants supporting the work. Thank you very much. Questions? Any additional questions? Yes.
Beacons can penetrate several layers of cells, or is there a very quick?
That's a good question. So depending on the delivery method you use, for the toxin-based method or slow-based method, usually you need to have a monolayer of the cells because if you have, let's say, a thin section of tissue, you have to use a very high concentration of slow in order to penetrate into a few layers, but that may kill cells. With the cell-penetrating peptides, actually, the peptide could lead the beacons to get into the first layer, actually come out to go into the second layer or third layer, right? So the answer is depending on what is the delivery method you use.
Then with the peptide, how deep do you think it can go?
So the longer you wait, the more layers they could go through. But I would think with a typical assay time, probably won't go beyond 5 layers. Right. But also, if you have more layers, you would also have the issue of light penetration and getting signal out. Yeah. Even if you use infrared dyes. Yes?
For photo bleaching, is there any way to reactivate the fluorescence?
With what?
Photo bleaching.
Oh, photo bleaching. In a way, yes. So Again, depending on the fluorophores, right? Some of the fluorophores, it could have a so-called dequench, but that may take a while, right? So although it's possible for most of the biological studies, perhaps you don't want to wait for too long, right? So I think a more effective way is trying to overcome photobleaching. Roger.
In the talks that we've seen so far from people who are doing work in developmental biology, looking at the early embryo,
Right.
We haven't seen a lot of fluorescent work.
Right.
With maybe the one exception where where you express a protein marker
Fluorescent protein.
of differentiation into a particular cell type. Is
Right.
there a reason for that? I mean, is this an opportunity for people?
Yeah. So most of the work so far with protein tagging, they try to use things like zebrafish, right, to study development because it's transparent, right? For some of the studies, unless the tissue itself is transparent, you would have the same issue, that is the limitation of light penetration, right? Also, there's a potential cytotoxicity if you shine light for too long, right? So there are different challenges. I think one of the major challenges is that in that case, you have to use things like fluorescent protein. And you want to follow the process, but you have photobleaching. Yeah, it's a challenge, yes.
What are the depths that you can realistically emit fluorescence to into the tissue?
So usually, if you use red dye or infrared dye, right, you can actually reach something between 10 to 100 microns in depth, right? So a cell perhaps in 3D, roughly 10 microns, right? And you could have like 5 layers without reducing the signal a whole lot, right? But once you go beyond that, you may still have some signal, but the signal would be pretty weak, right? Also, the deeper you go, the worse the spatial resolution, right? Yeah, so there are, again, challenges there, right? But I think that's an area we definitely want to do more work. Yeah.
If you're stuck with a fluorophore that photo bleach quickly, what are the best ways to minimize, maximize the use of it?
Right, so there I think you have several things. One is you could choose a dye which is more resistant to photobleaching instead of using a dye which photobleaches quickly. So that's the common approach people use, just to pick up a dye. Because it's really due to the interaction between the specific dye and the local biochemical environment. And in a cell, you cannot change the local environment. Therefore, the only way is to choose a dye. Actually, there are efforts now including in Roger Chan's lab, is to tailor or to chemically modify the dye so that it becomes more resistant to photobleaching. Right. Including, I know Roger Tian has been making some effort to modify the fluorescent protein so that it becomes more resistant to photobleaching. Right.
But there are ways you can change the biochemical environment to alter photobleaching.
Right. If you can alter the intracellular biochemical environment, right? If you can do that.
Yeah.
But sometimes there are also effects to the cell you don't want to have, right? For example, if you incubate cells with a specific type of buffer, right? Or the cell culture media, for example, right? And in some cases, you could reduce photobleaching, but you need to be careful there, not to alter the biology of the cell in doing so. Yeah.
What about extracellular probes?
Extracellular probe actually is actually a much better situation because for extracellular environment, you could easily alter the local environment so that the photobleaching would not be a major issue, right? And it's mostly challenging for intracellular studies.
Can you do things like alter the exposure time?
Yeah, you can use all kinds of tricks optically to, for example, some of the microscopes, like the one we use, called delta vision, it has a very nice engineering to make concerted exposure and taking images to reduce the exposure time, but still get a much better image, right? So as a way to reduce photobleaching, right? Yes, indeed. Yes.
You mentioned about the 50-fold difference between the editing.
Right, the DNA is single-stranded, the double-stranded. Right.
What would be an intuitive interpretation?
I'm sorry?
What would be an intuitive interpretation of that difference?
So why there's a 50-fold increase of that? Yeah, it's actually because of the binding strong electrostatic interaction between the two strands, right? And it's not, I would say, it's actually the increase of the binding rigidity people have studied a lot. It's not really due to the mechanical interaction between the two strands. It's mostly due to electrostatic interactions between the two strands. Yeah, to make the binding much stronger and make the strand, once it's double-stranded, make it much stiffer to resist deformation. Right? But a lot of actually detailed studies in that.
But you can't even explain that by the 8 to the 4th dependence of the bending of the moment of inertia. If it's only increasing by a factor of 2 in one dimension, that would only give you a factor of 16.
You cannot, exactly, that's why you cannot explain that by purely mechanical arguments, right? Because there's increased cross-sectional area and so forth. No, you cannot do that. Because that would give you maybe a few fold, but not 50-fold. Right, that's why, yeah. Yes?
Instead of using fluorescent, can we use other techniques like scattering and other?
Yeah, you can make use of scattering, right, for tissue imaging, right, like OCT and other methods. But those have some medical applications, but not really in terms of molecular imaging, because in that case, I haven't seen any engineered contrast agent for that, right? Yeah. So the limitations there. Okay, thank you.
So lunch will be